Welcome to the week 8 of the machine learning module. This week we have two topics. The first one is clustering and the second one is feature selection. The clustering lecture is split into two parts. In the first part we will describe k-means clustering and Gaussian mixture model, and in the second part we will talk about spectral clustering, and this builds on your knowledge of Laplacian eigenmaps from this module. So let's start with the first part. We will use Wisconsin breast cancer dataset as an example. So in this dataset, there were cells extracted through a fine needle aspirate, which is a type of biopsy, and then the digitized photograph was taken through a microscope. You can see the example of such image here. And then the cell boundaries are detected using snakes. This is an interactive method. Once we have boundaries of the cells, we can calculate various features of these cells. So there are 30 features in this data set. Examples are radius, texture, or area. Radius is just a mean of distances from the center to the points on the boundary. And then texture is standard deviation of the intensities in the cell, and area is number of pixels within the same cell boundary. So in each specimen, we have several cells, so we need to calculate some statistics. So types of statistics that are included is a mean value of all the cells, standard error, and worst value, which means largest value. There is also diagnosis available for these patients, which is malignant or benign. But because we talk about clustering, we will only use it for comparison, but not for training. So to start with that, we will select only two features so that it's easy to visualize: mean radius and mean texture in the first instance. And we already know how to classify to predict cancer diagnosis using the labels that are given in this dataset. But today we want to look at something else. We want to know whether there is natural structure in this dataset that we could discover without knowing the labels. So what do we mean? Is there something about healthy and cancerous cells that makes them fall into natural clusters within this dataset, that we could actually recognize them even without knowing what diagnosis the patient had? So this is done using clustering. And you already know that clustering is an unsupervised machine learning task, which means we don't have any training labels. And our aim is to split samples into groups according to some criterion. So what could be this criterion? So for example, we could minimize the within class variance, and this results in k-means clustering. So let's break it down what that means. So the sigma squares, that's our within class variance. And this within class variance is calculated from variances for each class. And these variances are weighted by the size of the class. So here you can see the number of samples within a class, and n is number of all samples. And we sum them up. So why would we do that? So let's say that we have two well-defined clusters. And if we split it like this nicely in the middle, and we have nice centers like that in the center of each cluster, obviously this is a good solution, and the variance within each class should be relatively small. But if we split it this way, this would not be a very good split, because obviously these parts of the cluster are very far from each other, so the variance will be large. So this is not a good split. So we are aiming at something like this. So how do we go about it? So we can understand how we could calculate this cluster means or cluster centers, or split the data into clustering, if we try to work out what this equation really means. So first thing is to write down what is the variance of each class, which we know very well. It's each sample minus the mean. So we can work on vectors. So this is Euclidean distance squared, and then we sum them up and divide by number of samples in that class. So that's the average distance from the center within the class. And then this can be simplified just to average distance of each sample from the class to which, from the center of the class to which it has been assigned. So intuitively, we can now see that the cluster centers and the clusters can be found by assigning a class with closest feature mean to each sample, and then recalculating the feature mean for each class after assignment. So you see in this video how that works. We start with random initialization, which is here. And then slowly we update. First we update the clustering, then we update the centers, and slowly it nicely converges to the correct cluster means and the correct separation into groups. So here is a result of k-means clustering for our breast cancer example with mean radius and texture of the cells. And here we can plot the boundary between the two clusters, and these are the original labels. So you see that we are not doing so badly, even though there are some misclassified samples. We have accuracy of 0.86 compared to the true labels. And if we apply the supervised method, such as logistic regression, we would get slightly better results. So you see that the decision boundary is a bit different, and we improve the performance to 0.89. But basically, the conclusion is that k-means done quite a good job actually finding the clusters just from the structure of the data without really knowing the true labels. So clearly there is structure in this data set. There is something about the cancerous cells that makes them different from healthy cells. Let's take another example. We now take a different pair of features, and it will be mean area and worst area. So that means largest area. And we see that the clusters are elongated in this case. And also it seems that these two features are quite strongly correlated. So k-means kind of works. We have accuracy 0.84, but the split doesn't look quite right. And I think what happened that k-means is not very good at modeling elongated clusters because we are only looking for distances from the cluster center. So let's see how Gaussian, so yeah, this arrow here points to this problem that we have with k-means. So let's see how Gaussian mixture model will do for this data set. And we can see now that this is much better. So actually it found this blue class, the benign cells very beautifully. There are just very few points that are missing. So it was able to detect this kind of elongated cluster. And we see that with the accuracy 0.93, that is quite high. And remember, this is without using the true labels. So this is just from looking at the structure of the data. So how did the Gaussian mixture model do this? So the main idea is that we assume that clusters can be modeled by Gaussian distributions. So you can see this example here. So here we have the original data. Now no labels are shown. This is just how the data set looks like. And a Gaussian mixture model can actually work out that there is one cluster here with this kind of very elongated Gaussian distribution, and second cluster like this. And this is the boundary of the between the clusters, and you see that this boundary is nonlinear. So Gaussian mixture model can actually detect elongated clusters with nonlinear boundaries. So how does Gaussian mixture model clustering work? So to simplify the things, we will move into an example in one dimension, which is segmentation of brain MRI. You've seen this example before. And in this case, features will be the intensities of voxels inside the brain. So each voxel is a sample, and there is only one feature per sample, which is the intensity in the image. And we don't consider outside of the brain in the image. This outside is padded by zeros. So these are not features. And if you look at the histogram of this image, we can see that it has three nice peaks, and these peaks correspond to three tissues. The darkest one is cerebrospinal fluid, then the middle one is gray matter, and the brightest one is white matter. And this is the corresponding segmentation or clustering result. So as we said, the assumption is that intensity distribution for each class or cluster K is Gaussian. So you see here the peaks in the histogram and nice Gaussians that approximate these peaks. So this is my notation for a Gaussian distribution. You see that we have, this is the intensity, this is the mean, and this is the standard deviation. This is the standard equations for the Gaussian distribution. And this Gaussian distribution for each class is actually a probability of observing certain pixel or voxel intensity given the label. Okay, so for each pixel and its intensity, we can calculate this kind of probability that it's from class K. And this probability is expressed by these three different Gaussian distributions. So now we would like to express the probability of intensity, observing an intensity independent of which class is it. And we can model that as a mixture of Gaussian distributions. So we basically just create a mixture of probability distributions for each class. So we have here this extra parameter, mixing proportion, and we sum that up to get the probability. Now, if you look at the histogram, what we do, we are just making a combination of these probability, Gaussian probability density distributions to fit the whole histogram. So that's our aim. So when I talk about histogram, actually, to be completely precise, it's normalized histogram because it really needs to be a probability density function, and area of this density function under the curve needs to add up to 1. So to get a probability density function from histogram, we have to divide it by number of samples, and then we will have this property fulfilled. So now we know that Gaussian mixture model has 3 parameters per class or per cluster. So mean, standard deviation, and mixing proportion. And the question is, how do we find these optimal parameters so that our Gaussian mixture model fits the image intensity distribution as well as possible. And to do that, we will maximize log likelihood of observing our particle image. So this image X is something we know, parameters is something we don't know, and we are looking for the parameters that could create this Gaussian mixture model that best explains observing our image. So this is our likelihood, and you see we take logarithm of it in a minute. So likelihood is just a probability of observing the image that we've got given the Gaussian mixture parameters. And if we assume that the voxel intensities are independent from each other, we can rewrite it like this. So you know that we would multiply the probabilities for each voxel, and if we take logarithm, then it's actually just sum of logarithms of these probabilities. And here we got our Gaussian mixture. So likelihood for an individual pixel or voxel intensity is our Gaussian mixture. So here you can see two different examples of Gaussian mixture model. And this one you can see doesn't fit the intensity distribution of the image very well. So this will have a low likelihood, and this one fits it very well. So this will have high likelihood. Okay, so now we have our objective to maximize. So to maximize that, we now have to set the first derivatives with respect to the Gaussian mixture parameters to 0. And if we do that, then we'll get this type of equation. So first we have the equations for the Gaussian mixture parameters, but there is something PIK that suddenly appeared here. And this PIK is the probabilistic prediction. So it's the probability that for pixel intensity xi, we will have label k. So the opposite way as likelihood. This is called posterior. It's opposite to likelihood. And this is the expression. So you see how we calculate it. And actually, these probabilistic predictions in the context of image segmentation can be interpreted as probabilistic segmentations. So you see we have nice probabilistic segmentation for our 3 tissues. And then we will again have iterative algorithm. We will iterate between estimating the parameters phi using this set of 3 equations and recalculating the probabilistic segmentations PIK, or in a more general case, probabilistic predictions of cluster labels. And we do that until convergence of log likelihood. So here is the movie showing how this algorithm can fit the Gaussian mixture model into the distribution of intensities of a particular image. And you see how we start with something that's not fitting very well and it's progressing into a good fit. And also if you look at the probabilistic segmentation, they don't look very nice at the beginning, but slowly converge to a good solution. So once we have this final probabilistic segmentation, we can calculate the labels by assigning a label with highest probability to each voxel. So here is the code, how we implement clustering in scikit-learn. So you already know how to implement k-means. You've seen this object before. You see that we need to set the number of clusters that we expect. And we can use function fit predict that actually will fit the model and predict in the single command. And the reason that we can do that is because we are not really fitting to the labels. So there is no need to split into training or test set or doing cross-validation. Similarly, we also have Gaussian mixture, and we have to set the number of components, and then we can use fit predict. Here you can see the results of k-mean clustering and Gaussian mixture model for our first example. And you see that k-mean clustering results in linear boundary between the clusters, while Gaussian mixture model results in nonlinear boundary. So this is the end of the first part. Now please do the quiz on KISS that is called clustering. And once you are done with the quiz, please watch the second part on spectral clustering.